Hello students, welcome back to another new video of educators on digestion of fat. So we have done digestion and absorption of carbohydrate and protein, and now today we will discuss about how fats are digested. So in the picture only you can see that there are so many types of fatty foods all over. Some are known as trans fat, saturated fat, unsaturated fat, and over here we have shown the digestion of fat in a very simple three-step picture or image, like clumped fat, separated fats, and then we are getting fatty acids. So fats is basically a non-polar compounds which are chemically the esters of fatty acids and glycerol. We all know that. Now how this fat that we eat are digested in our body, that we will know through this video. Welcome to Educators, myself Manjishta Chatterjee. Don't forget to like, share and subscribe my channel. First, we must know why do we need fats? Because fats help brain development. That is 60% of the brain is fat. So some good fatty acids. I hope you have seen the advertisement of Horlicks where DHA, EPA has been mentioned. So what is this DHA? What is this EPA? So it is docosahexaenoic acid and eicosapentaenoic acid, which are very essential fatty acids important for brain development of the child. So fat doesn't always mean that it is very bad or it is harmful for our body. There are certain fats which are definitely harmful for our body, but there are also fats which are good for our body. Fat are also a structural component of cell. It is very important. So you know the protein lipid protein layer or the PLP layer. So that lipid layer is what the fat is, and it is a very important structural component of the cell. We know that there are certain vitamins like vitamin A, D, E, K, which is absorbed in our body only with the help of fat. So fat is important for the absorption of these vitamins. There are certain types of fat like the omega 3, omega 6, which help us to maintain a healthy heart and blood vessels. Definitely, it is also a source of energy. Fat provides 9 kilocalorie of energy per gram. So fat is essential for that as well. So now we will come to know that the type of food that we eat, the fatty food, how it is digested in our body and how it does get, um, how are we getting the energy or the different functions of the fat. The main types of lipids that we have are triglycerides, phospholipids, and steroids and waxes. So these are the basic type of lipids that we have. We either eat fat as good fats, good sources of fats, or as bad fats. In the introduction slide also, there were certain pictures. So those are the sources of fats in our diet, which is very, very important. Now the main or the basic structure of the fat is this triglyceride. Okay, we have three molecules of fatty acid, 1, 2, 3, and one molecules of glycerol, which is very, very important. Now what we need is that with the action of this lipase enzyme, lipase is the fat digesting enzyme. So with the action of this lipase enzyme, this triglyceride are gradually divided or digested into free fatty acid and glycerol. Okay, so first the triglyceride is mm, digested into diglyceride and free fatty acid, then to monoglyceride and free fatty acid, then to free fatty acid and glycerol. So this is the way, or this is step by step how fat is digested by lipase enzyme. Along with that, with to help the fat digestion, there are also certain things in our body which we will learn in detail in the next slides. So this is a nutshell of the digestion of fat. We know that saliva doesn't contain any lipase. So there is no fat digestion that occurs in the mouth. But like the other system, teeth and our tongue and the saliva, they help in the breaking of the fat into smaller particles and also masticating. So digestion of fat doesn't start, but the process of digestion, like the chewing and masticating, that process starts in mouth. Coming to the gastric lipase, which is present in the stomach, the fat digestion starts a bit with the gastric lipase. So over here we'll come to this picture. We can see that this is a fat droplet which is emulsified. Now what is emulsion, we will come to know. This is a fat droplet which is emulsified, and gastric lipase acts on this emulsified droplet. Always keep in mind that lipase enzyme can work better in a emulsified fat. Okay, it works better. Now what happens with the action of this gastric lipase? The large fat molecules, so these are the gastric lipase, the violet colored one. Okay, they are actually going and getting itself attached to the surface of the fat molecules, the large fat molecules, and they help in the breakdown of them into smaller particles. Okay, they are breaking down into smaller particles. So up to this what happens in the stomach? The stomach does not have any much greater role, but the stomach lipase can act up to this point of action. But always keep in mind that the stomach can only act at the 40 degrees centigrade. This is the optimum temperature of the activity, and this gastric lipase acts good or acts better in acidic medium, but it can also uh, be denatured under the higher action or excessive action of the HCl. So if the acidity or the HCl action is too much, then the gastric lipase's function will be denatured. Okay, so it will break up to 1 molecule of glycerol and 3 molecules of fatty acid. Now next what happens, we can see this fat moves into the small intestine. As soon as it moves, the fat will be getting bile, receiving bile, and it will change the emulsified fat. So once bile is mixed with the fat molecules, they will be much more, they will be broken into smaller particles, and they will be emulsified so that the action of the lipase enzyme is better. Then on this emulsified fat, the pancreatic lipase acts, and they are gradually digesting them finally into monoglycerides and fatty acid. So over here we can see that this pancreatic lipase, they bind to the fat droplets, and they gradually are getting like this, which are forming micelles, and finally they are moving to this microvilli for absorption. So small intestine over here, what happens? See, the acidic stomach content, which has chyme, which we have discussed in the previous digestions, that what is a chyme? They leaves with the content of the dietary fat, they leaves the stomach and they enters into the small intestine. As soon as the fat containing chyme, they enters into the small intestine, there is a secretion of the secretin and cholecystokinin, or the CCK. What happens? What happens when the fat enters? The gallbladder is acted upon, which helps to release the bile. Okay. It also acts upon the exocrine cells of the pancreas, and they release the pancreatic lipase. And further, there is another function, the gastric motility. Okay, Antiperistalsis actually takes place over here in the duodenum. The gastric motility lowers, and which increases the time for digestion and content. You will see that if you eat a fruit, you feel hungry, but if you eat a burger, you don't feel hungry so much, right? Why? Because the satiety value of fat is high, and the fullness value of fullness is also of fat is very high, because of this antiperistaltic movement in the duodenum. Okay, because it takes time to get digested, which is another which is very good, because as it spreads and a little bit in a little bit way, they are getting digested in the small intestine. So which are the fat busters? First is the secretin, which is a peptide hormone. It helps the or tells the liver to make more bile. So whenever there is any fatty food in your stomach, or fatty food in your mouth rather, the secreting is produced, and this peptide hormone tells the liver to make more bile so that there is better fat absorption. And there is lipase. Lipase also is the enzyme which breaks down fat. I've already discussed about the CCK, or the cholecystokinin, which actually tells that the chyme has the, if the chyme has the fat, then the CCK helps, or CCK hormone tells the gallbladder to release the bile. And bile, when the gallbladder releases its contents, bile salts in the bile, they reach the duodenum, and they make the surface very very huge surface, so that absorption is good, and it mixes with the fatty products as the with watery surfaces to act like a detergent, and it cleans up, so it aids the digestion process. So now, coming to the function of the bile salts, which is very important, I just discussed about the emulsification, which is very important action or function of the fat. So this is a fat globule, and bile salt always have two sides. One is the hydrophilic side, that side loves the water, and a hydrophobic side, which is feared of water. Okay, so what happens, the fat globule, the hydrophobic sides, they get attached to the fat globule and make them smaller fat globules with bile salts all over it. So this is how the thing looks like. Okay, so definitely it enhances the absorption of fatty acid and also the fat soluble vitamin. It helps in emulsification by reducing the surface tension and breaking the fat molecules into smaller particles. And also, when the emulsification is done, the fat molecules they spreads, and the little molecules can be acted upon great by the lipases. Also, fat has bile has a cholagogue action. Cholagogue action means it when the bile is needed, it generates, or it synthesizes itself. Okay, so that's the cholagogue action of the bile. Okay, and also bile maintains the pH of the the pH of the system, so that the action of the lipase is much better on the fat, and the fats get digested in a proper way. So after, this is what happens in the small intestine. So one part is always regulated by the bile salts of fat digestion in the small intestine. So one part, as I was discussing in the previous slide, that one part was definitely controlled by the bile salts, the bile helping in emulsification and further action of the lipase, and the second part of the digestion takes place by the lipase enzyme, which is present in the small intestine. One is the cholesterol ester, another is the cholesterol ester hydrolase, and the phospholipase. Phospholipids respectively acts on the phospholipid, forming into lysolecithin and fatty acid, whereas cholesterol acts on the cholesterol ester and they form the cholesterol and fatty acid. So what we want is by the triglyceride, by the lingual, gastric and the pancreatic lipase to convert into monoglycerides and fatty acid. So this is the basic or a nutshell of the digestion of lipid. Digestion of lipid also requires certain other factors too. Okay, before that, let me tell you that succus entericus does not help in maximum of the digestion of fat takes place in the stomach and the small intestine rather, but succus entericus does not take any part in the fat digestion. Now coming to the different factors which affects um, fat digestion is first is the entrogastrone. This entrogastrone, as I was discussing, the slowing down of the digestion of fat in the stomach. So entrogastrone helps in the peristalsis and acid secretion of the stomach, and this slows the digestion in the stomach so that the fat digestion can be eventually done slowly and gradually and properly. And antiperistaltic movement, which occurs between the stomach and the duodenum, and it allows more time for neutralization of the chyme in between the cecum and the ileum, so that the absorption time increases. Okay, other than that, arginine, histidine and lysine, these three amino acids also help in the fat digestion. So after a fat is digested, after a fat is digested, at the end of this digestion they are broken into fatty acid and glycerol, and some amount of monoglycerides may be present. All these compounds are bound up together to produce micelles, which is moving forward to the absorption of fat. So in the next slide, in the next presentation, we will be discussing about the absorption of fat. I hope digestion of fat is clear to you. Next presentation will be discussing about absorption of fat. Stay tuned with educators. Thank you.